Hello there and welcome back to Eternal Cosmos, where wonder takes its time and questions don't demand immediate answers. Tonight we're going to do something that shouldn't be possible. We're going to visit a day that happened before days had witnesses, before eyes existed to see sunrises, before there was anything alive to mark the passing of time. We're going to find our way back to the moment when chemistry became biology, when Earth stopped being just a planet and started being a home. But here's the thing about beginnings. They're almost never dramatic. The first spark of life didn't announce itself with fanfare. There was no sudden flash, no divine breath, no moment when the universe took notice. Instead, somewhere in the chaos of early Earth, in water that would have killed us instantly, under skies that had never known oxygen, something shifted. A molecule learned to make copies of itself. And though it would take billions of years for that copying to become cells, consciousness, and eventually you, lying here listening to this story, that first moment was already everything, the seed of every living thing that would ever be. Before we travel back those 4 billion years, I'd love to know where you're listening from tonight. What time zone cradles you right now? It's funny, isn't it? Here we are, scattered across a planet that once knew no life at all, each of us carrying within ourselves the memory of that first moment. We're all descended from that original spark, that first successful experiment in being rather than just existing. If this kind of deep time wondering speaks to you, if you find yourself curious about the immense story that preceded us all, I'd be grateful if you'd consider subscribing or leaving a like. It helps this small voice reach other wondering minds. But no pressure, just an invitation, like the soft pull of gravity that once gathered the dust that would become our world. So settle in, let your breathing slow, let your thoughts drift back, not just through human history, not just through the age of dinosaurs or the first fish, but all the way back to when Earth itself was young, violent, and completely unaware that it was about to become the only known address for life in the universe. Part 1. The Hostile Cradle. 4.5 billion years ago, before we begin our story properly, Earth wasn't Earth yet, not in any way we'd recognize. It was a collision still cooling, a gathering of debris still deciding what it wanted to be. The solar system itself was barely organized, still full of wandering rocks, leftover pieces from the sun's formation, chunks of metal and ice, following wild orbits that sometimes intersected with devastating results. Picture this early Earth. No blue marble suspended in the void, no green and brown continents, no white clouds. Instead, imagine a world of black rock and red magma, a surface that couldn't decide if it was liquid or solid. The planet glowed with its own heat, not from sunlight warming its face, but from the violence of its own formation. Every few minutes, another meteor would slam into the surface, adding its mass to the growing world, keeping everything molten, keeping everything chaos. The moon, if you could call it that yet, hung much closer than it does now, maybe 10 times closer. It loomed enormous in the sky, pulling at the barely formed Earth with such force that the rock itself responded like a tide. Imagine solid ground rising and falling by hundreds of feet, the entire planet flexing like a living thing, though nothing living had yet appeared. And the days were so brief. Earth spun like a desperate dancer, completing a full rotation in just 6 hours. 6 hours from sunrise to sunrise, if there had been anyone to see the sun through the thick envelope of gases surrounding the planet. But that envelope wasn't air, not yet. It was methane, ammonia, water vapor, carbon dioxide, a toxic soup that would have killed any modern life in seconds. There was no oxygen. That would come much, much later, and when it did, it would be poison before it became essential. The sun itself was different then, younger, weaker, about 30% dimmer than the star we know today. It was still settling into its rhythm, still finding its balance between the outward pressure of fusion and the inward pull of gravity. Its light, such as it was redder, softer, but you wouldn't have seen it anyway. Not through the thick atmosphere, not through the constant volcanic ash, not through the steam rising from endless impacts. Because Earth was still being built, even 500 million years after the solar system began forming, pieces were still falling into place. The late heavy bombardment, that's what we call it now, though late is relative when you're talking about the first billion years. Asteroids, comets, planetesimals, they all took their turns adding mass to Earth, and each impact was catastrophic by today's standards. Some of these weren't small rocks, Some were the size of cities, mountains, small countries. When they hit, they didn't just make craters, they vaporized. They turned solid rock back into gas, threw molten debris thousands of miles, sometimes into space. The energy released could melt entire regions, keep them liquid for thousands of years. And yet, this violence was necessary. It was delivery as much as destruction. You see, many of these impactors, especially the comets, carried water, ice that had formed in the cold outer reaches of the solar system, now being delivered to the inner planets. Each impact was hell, but it was also rain waiting to happen. Water that would eventually cool, condense, and become the oceans where life would begin. But not yet, not for millions of years yet. The atmosphere during this time was dense, crushing. The pressure at the surface might have been 100 times what we experience now. It was more like being at the bottom of an ocean than standing on land, and it was hot. Not just from the impacts and volcanism, but from the greenhouse effect of all that carbon dioxide and methane. Even with a dimmer sun, the surface temperature could reach 400 degrees Fahrenheit, sometimes higher. Hot enough that if water did briefly condense, it would immediately flash back to steam. And the geology, if we can even call it that when everything was so fluid, was nothing like the rigid plates we know today. There were no continents, no ocean basins, just a kind of rocky foam, constantly churning, constantly recycling itself. Heavy elements sank toward the core, lighter ones rose. The planet was differentiating, organizing itself into layers, but it was a slow, violent process. Iron and nickel, being dense, descended through the molten rock like drops of mercury through water. They gathered at the center, forming what would become Earth's core. This wasn't a gentle settling. As these massive amounts of metal fell toward the center, they released enormous amounts of energy, enough to melt the entire planet several times over. This is called the Iron Catastrophe, and it was exactly as dramatic as it sounds. The formation of the core did something else crucial. It set up the conditions for Earth's magnetic field, though that wouldn't fully develop for perhaps another billion years. But the seeds were there, a rotating ball of liquid metal that would eventually generate the invisible shield that protects us from solar radiation. Without it, life as we know it couldn't exist. But again, not yet. These are still the planet's youngest days, when everything was potential and nothing was certain. Meanwhile, the surface was a landscape of volcanoes, not the isolated peaks we think of today, but entire regions of volcanic activity. Imagine thousands of Yellowstones all active at once, lava lakes the size of small continents, fountains of molten rock shooting miles into the atmosphere, and between them, when things briefly cooled, a surface of black basalt, cracked and steaming, looking like the skin of some impossible creature. The moon hanging so close would have been a constant presence if you could have seen it through the murk. But it too was cooling from its own violent birth, likely the result of a Mars-sized object called Theia colliding with Earth, perhaps 100 million years earlier. The debris from that impact had coalesced into our satellite, but it was still glowing with heat, still covered in its own magma ocean. Two molten worlds, locked in gravitational embrace, slowly cooling together. And through all of this violence, this chaos, this seemingly impossible environment, chemistry was happening. Not life, not yet, but the precursors to life, the building blocks. Because even in this hell, even in conditions that seemed designed to destroy rather than create, molecules were forming, breaking, reforming. Simple became complex, random became patterned, and somewhere in that chemical chaos, the stage was being set for the most important moment in Earth's history. But we're getting ahead of ourselves. First, the planet had to cool. First, the impacts had to slow. First, water had to condense. First, the surface had to become something other than an endless expanse of lava and ash. And that would take time, millions of years, tens of millions. But time was something the young Earth had in abundance. Part 2. When water first fell. Imagine standing on Earth 4.4 billion years ago. Actually, you couldn't stand. The surface would either be molten or, in the rare solid spots, hot enough to cook you instantly. But imagine observing somehow as the planet underwent one of its most profound transformations, the day the rains began. It didn't start as rain, not really. For millions of years, water existed only as vapor in that thick, crushing atmosphere. Every time a comet delivered more ice, it would vaporize on impact. Every time volcanic eruptions brought water from the planet's interior, it would become steam. The atmosphere was saturated, supersaturated, holding more water than all of today's oceans, but holding it as gas in skies that glowed red with heat and dust. But planets cool. It's what they do. Slowly, inevitably, they radiate their heat into space, and as Earth cooled, degree by degree over millions of years, it approached a critical threshold, the temperature where water vapor could condense into droplets, where those droplets could fall, where rain could exist. The first drops wouldn't have made it to the surface. They would have formed high in the atmosphere, fallen a certain distance, then evaporated again in the heat below, like ghost rain, never quite reaching the ground. This might have gone on for thousands of years, millions, a constant cycle of condensation and evaporation, water falling and rising, never quite completing its journey. But gradually, inexorably, the surface cooled enough. The atmosphere thinned slightly as gases escaped to space or were absorbed into rocks. And one day, though day is a strange word when days lasted only 6 hours, one day, the first drops made it all the way down. They would have hissed and steamed on contact with the still hot rock. They would have vanished almost instantly, but more followed, and then, perhaps triggered by some volcanic winter or a pause in impacts, the deluge began. Try to picture rain like nothing in human experience, not a storm that lasts hours or days, but rain that continued for centuries, millennia, maybe millions of years. The entire atmospheric reservoir of water, finally able to condense, falling in sheets so thick you couldn't have seen through them. The sound would have been deafening, a constant roar of water meeting rock, water meeting water, the birth cry of the oceans. And what rain it was! This wasn't the relatively pure water we know. It was acidic, loaded with dissolved carbon dioxide, sulfur compounds, chlorine. It would have etched the rocks it touched, chemically weathering them, breaking them down, creating the first sediments. The rain was literally dissolving the planet's surface and remaking it. As water accumulated in the low places, and there were many impact craters, collapsed lava tubes, depressions between volcanic peaks, it didn't form oceans immediately. First came pools, then lakes, then small seas, probably temporary, repeatedly evaporated by volcanic heat or new impacts, then refilled by the endless rain. The temperature was still brutal. These early bodies of water were hot, not boiling everywhere, but warm enough to kill any modern life. Think of them as planet-wide hot springs, rich in dissolved minerals leached from the rocks, iron, magnesium, silicates, all the elements that had been locked in stone now entering solution, creating a chemical soup of extraordinary complexity. And the color, these weren't blue seas. The early oceans were probably green from dissolved iron, or rusty red, when that iron oxidized, or brown with suspended sediments. They were murky, opaque, more like a witch's cauldron than the clear waters we associate with tropical beaches. But they were water, liquid water, the universal solvent in which chemistry could happen at speeds impossible in solid rock or diffuse gas. As the oceans grew, they began to change the planet in fundamental ways. Water has an enormous heat capacity. It can absorb vast amounts of energy without changing temperature much. This meant the oceans became a thermal buffer, moderating the planet's temperature, smoothing out the extremes. Days became slightly less scorching, nights slightly less frigid. The water was teaching Earth how to be stable. The moon, still so close, pulled at these new oceans with incredible force. Tides weren't the gentle twice-daily rises we know. They were massive, violent movements of water. Imagine tides hundreds of feet high, racing across the landscape at tremendous speeds. In 6-hour days, with the moon so close, these tides would have been relentless, constantly stirring the waters, constantly mixing chemicals, constantly creating and destroying coastal environments. This tidal mixing was crucial. It prevented the oceans from stratifying into layers. It ensured that minerals from the seafloor were constantly cycled upward, that atmospheric gases were mixed downward, that no part of the ocean could become isolated from the grand chemical experiment taking place. The moon, through its gravitational pull, was stirring the pot from which life would emerge. Meanwhile, underwater, something remarkable was happening. Hydrothermal vents, cracks in the seafloor where water met hot rock, were creating oases of chemical activity. Cold seawater would sink into these cracks, be heated by magma below, dissolve minerals from the rock, then surge back up, laden with dissolved metals and gases. Where this hot, mineral-rich water met the cooler ocean, precipitation would occur. Minerals would crystallize out of solution, creating chimney-like structures, some towering dozens of feet above the seafloor. These weren't like modern black smokers, though those are their descendants. The chemistry was different because the ocean chemistry was different. No oxygen meant different minerals could stay dissolved. The pH was different, the temperature gradients were more extreme, but the principle was the same. A boundary between two different chemical environments, a place where energy flowed and reactions could happen that were impossible elsewhere. Some scientists now think these vents were crucial for life's origin, not just because of the chemicals they provided, but because of the structures they created. The mineral chimneys weren't solid, they were porous, full of tiny chambers and channels. Each microscopic chamber was like a natural test tube, a place where chemicals could concentrate, reactions could proceed without being diluted by the vast ocean. They were natural laboratories, running millions of experiments simultaneously. And in the water itself, other processes were at work. Ultraviolet light from the sun, unfiltered by an ozone layer that didn't yet exist, was breaking apart simple molecules and causing them to recombine in new ways. Lightning, and there was plenty of lightning in that turbulent atmosphere, was providing jolts of energy that could forge chemical bonds impossible under normal conditions. Radioactive decay from elements in the rocks and water provided a steady source of energy. Even the impacts that continued, though less frequently, delivered both organic compounds and the energy to transform them. The oceans became a vast chemical reactor. Every possible combination of elements was being tried somewhere. Most led nowhere. Unstable molecules that broke apart immediately. But some persisted. Some even catalyzed the formation of more molecules like themselves. The ocean was learning chemistry, trying every possible combination, keeping what worked, discarding what didn't. Carbon was especially versatile. It could form long chains, branch structures, rings. It could bond with hydrogen, oxygen, nitrogen, sulfur, phosphorus. The variety of carbon-based molecules, organic molecules, that could form was essentially infinite. And in the warm, mineral-rich waters of early Earth, many of them did form. Amino acids, the building blocks of proteins, simple sugars, fatty acids that could form membrane-like structures, nucleotides, the components of RNA and DNA. But forming these molecules was one thing. Organizing them into something that could be called alive, that was something else entirely. That required not just the right chemicals but the right conditions, the right energy flows, the right containers, the right catalysts. It required something that could maintain itself against the tendency of everything to fall apart, something that could make copies of itself, something that could evolve. That something was coming. But first, the planet had more preparing to do. The oceans had to stabilize. The temperature had to find a range that could support complex chemistry. The bombardment had to slow enough that large impacts wouldn't repeatedly sterilize the surface. And perhaps most importantly, there had to be places, special places, where all the necessary conditions came together. Part 3. The Chemical Symphony. In the warm, mineral-rich waters of early Earth, something extraordinary was happening. Not life, not yet, but the rehearsal for life. A vast chemical symphony where every molecule was both instrument and composer. Every reaction a note in a composition that had been playing for millions of years without an audience. Let's talk about carbon for a moment. Not in the way chemistry textbooks do, with their neat diagrams and electron shells, but as the protagonist of our story. Carbon is promiscuous. Chemically speaking, it forms bonds eagerly, diversely, creatively, while silicon, its neighbor on the periodic table, tends toward rigid crystalline structures. Carbon is flexible, playful almost. It makes chains that can be straight or branched, rigid or flexible. It makes rings that can be flat or puckered. It can form single bonds, double bonds, triple bonds, each changing the character of the molecule entirely. In those early oceans, carbon was everywhere. Dissolved carbon dioxide from the atmosphere, methane seeping from hydrothermal vents, simple organic molecules delivered by comets and meteorites. And in the presence of energy, lightning, UV radiation, volcanic heat, these simple carbon compounds began combining into more complex forms. The Miller-Urey experiment, performed in 1952, gave us a glimpse of what might have been happening. Take water, methane, ammonia, and hydrogen. gases thought to be present in Earth's early atmosphere. Add electrical sparks to simulate lightning. Within days, amino acids form, the building blocks of proteins, created from simple gases and energy. It seemed almost too easy. Later experiments showed it was even easier than Miller and Urey thought. You didn't need exactly their mixture of gases. Almost any combination of simple carbon compounds, water, and energy would produce organic molecules, amino acids, nucleotides, simple sugars. They all formed readily under a variety of conditions. The early Earth was a natural organic chemistry laboratory, running countless experiments in parallel. But here's where it gets interesting, and also problematic. Making organic molecules is easy. Making them work together in organized ways is incredibly hard. It's like having all the letters of the alphabet, but no grammar, no words, no meaning. You can scatter letters on a page all day, but they won't spontaneously organize into Shakespeare. This is where concentration becomes crucial. In the open ocean, even if complex molecules formed, they'd be immediately diluted. Imagine dropping a sugar cube into the Pacific. The molecules disperse, drift apart, never encounter each other again. For complex chemistry to happen, molecules need to be crowded together, forced to interact. They need what chemists call molecular intimacy. Nature provided several solutions: tide pools that concentrated chemicals as water evaporated, ice, which excludes dissolved substances as it freezes, creating pockets of concentrated brine, clay surfaces that could absorb organic molecules, holding them in place while they reacted, and those hydrothermal vents with their mineral chambers, each one a tiny reaction vessel. But the most elegant solution might have been the simplest bubbles, or more precisely, microspheres, tiny droplets of oil in water, or water in oil. You see, some of the organic molecules forming in the early oceans were fatty acids, long chains of carbon with a water-loving head and a water-hating tail. Put enough of these in water, and they spontaneously arrange themselves into spheres, with the water-hating tails pointing inward and the water-loving heads facing out. These weren't cell membranes, not yet. They were simpler, less selective, less stable, but they created an inside and an outside. They created a boundary between self and environment, and that boundary, that simple distinction between in here and out there, might have been one of the most important inventions in the history of Earth. Inside these microspheres, chemistry was different. Molecules could concentrate, reactions could proceed without interference. If a useful molecule formed, it stayed local instead of drifting away. And if these spheres happened to engulf the right combination of molecules, say, some amino acids, some nucleotides, maybe a clay particle that could act as a catalyst, then you had a tiny, isolated experiment in organization. Millions, billions of these experiments were running simultaneously. Most led nowhere. The spheres would form, persist for a while, then break apart. But some might have been special. Some might have contained molecules that could speed up certain reactions. Some might have been better at absorbing useful materials from their environment. Some might have even been able to grow and divide, splitting into two spheres when they got too large. This wasn't life. There was no heredity, no evolution in the Darwinian sense. But it was organization. It was complexity maintaining itself against the universal tendency toward disorder. It was a step towards something more. Meanwhile, another crucial development was taking place. Certain molecules were discovering that they could serve as templates for making copies of themselves, not perfect copies. Errors were common, but copies nonetheless. RNA, it turns out, is particularly good at this. Unlike DNA, which primarily stores information, RNA can both carry information and catalyze reactions. It can be both the blueprint and the builder. The RNA world hypothesis suggests that before DNA and proteins, RNA molecules might have been the first self-replicating entities. In the laboratory, we've created RNA molecules that can copy themselves without any protein enzymes. They're slow and error-prone, but they work. And in the vast chemical laboratory of early Earth, with millions of years to experiment, even slow and error-prone could eventually lead to something remarkable. Imagine an RNA molecule that could make rough copies of itself. Most copies would be worse than the original, less stable, less capable of copying. They'd fall apart and disappear. But occasionally, by pure chance, a copying error would create something better, more stable, faster at copying, better at grabbing the necessary building blocks from the surrounding soup. That improved version would make more copies of itself, and its descendants would become more common. This is evolution at its most basic. No cells, no organisms, just molecules competing for resources, and the ones that copy themselves most successfully becoming more abundant. It's so simple it seems almost inevitable. Given self-replicating molecules and limited resources, evolution must occur. The molecules have no choice in the matter. They're just following the laws of chemistry and mathematics. But naked RNA molecules in the ocean would face challenges. They'd be broken apart by the very energy sources that created them. They'd be diluted in the vast waters. They'd struggle to find the building blocks needed for replication. This is where those microspheres become crucial. Again, an RNA molecule inside a fatty acid bubble would be protected. It would have a controlled environment. And if that RNA molecule could do something useful for the bubble, maybe produce a molecule that made the membrane more stable or helped it grow, then you'd have the beginning of cooperation between container and content. This is starting to sound like life, isn't it? A boundary defining self from non-self. Information molecules that can replicate with variation. A cooperation between different chemical systems. But we're not there yet. These early systems, whatever they were, would have been incredibly fragile. A slight change in temperature, or pH, could destroy them. They had no way to actively maintain themselves. No metabolism in the true sense. They were more like chemical patterns that could sometimes persist and spread. But patterns have a remarkable property. They can build on each other. A pattern that helps preserve another pattern will be preserved along with it. RNA molecules that could instruct the formation of useful proteins would be more successful. Proteins that could protect and assist RNA would likewise thrive. Membranes that could selectively allow materials in and out would give their contents an advantage. Slowly, through countless random experiments and the ruthless filter of selection, complexity would accumulate. The key insight is that this didn't happen all at once. There was no moment when chemistry became biology. Instead, there was a long, gradual transition. Chemical systems becoming more lifelike, lifelike systems becoming more complex. Each tiny improvement opening the door to the next. It might have taken hundreds of millions of years, this slow climb from simple chemistry to the first things we could truly call alive. And it might have happened more than once. Different chemical systems might have achieved lifelike properties independently. They might have competed, cooperated, or merged. The early history of life might have been more of a network than a tree, with different lineages sharing innovations, stealing useful molecules from each other, combining and recombining in ways that would be impossible for modern organisms. We don't know exactly how it happened. We may never know. That information is almost certainly lost, erased by the very success of the life that followed. But we can see echoes of it in modern biochemistry. The universality of certain molecules, the deep conservation of certain chemical processes, the fact that all life we know of uses the same basic chemistry suggests a common origin, a single solution that worked so well it eventually dominated everything else. Part 4. The architecture of membranes. Think about your skin for a moment, not in the way you usually might, as something that tans or wrinkles or needs moisturizing, but as a boundary. It keeps your insides in and the outside out. It lets some things through, oxygen, water vapor, while blocking others. It maintains a difference between you and everything else. Without it, you dissolve into your environment, literally. Now imagine the first boundary, not skin, not even a cell membrane as we know them today, but the first successful attempt to create a persistent difference between here and there, between inside and outside. This might have been the most important architectural innovation in the history of Earth, more fundamental than DNA. More crucial than proteins, because without a boundary, there can be no self, and without self, there can be no life. In the warm waters of the early ocean, fatty acids were accumulating. These simple molecules, just chains of carbon and hydrogen with a carboxyl group at one end, had a peculiar property. One end loved water, the other hated it. Put enough of them in water, and they'd organize themselves spontaneously, trying to satisfy both preferences at once. The water-hating tails would cluster together, while the water-loving heads faced outward toward the water. At low concentrations, they might form simple micelles, tiny spheres with the tails pointing inward. But at higher concentrations, something more interesting happened. They formed bilayers, double sheets with tails facing tails in the middle, heads facing outward on both sides. And these sheets, being flexible, would curve and eventually close on themselves, forming hollow spheres, vesicles, protocells, the first draft of every living cell that would ever exist. These early membranes were nothing like the sophisticated barriers that surround modern cells. They were leaky, unstable, prone to falling apart. But they had one crucial ability: they could grow. As more fatty acids were added from the environment, the membrane would expand, and when it got too large, mechanical forces would cause it to divide, pinching off into two smaller spheres. No complex machinery needed, just physics. But here's where it gets beautiful. These membranes weren't just passive containers. They were selective, in a crude way. Small molecules could pass through relatively easily, large ones couldn't. Charged molecules had a harder time than uncharged ones. This meant that the inside of a vesicle could have a different chemical composition than the outside. It could be its own little world, with its own chemical rules. And if something interesting was happening inside, say an RNA molecule was replicating, or a chemical reaction was producing useful compounds, that activity was contained, protected, and able to proceed without being diluted in the vast ocean. The membrane wasn't just a bag, it was a laboratory bench, a reaction vessel, a proto-organism. Modern cell membranes are made of phospholipids, more complex molecules than simple fatty acids, with a phosphate group that provides better stability and control. But experiments have shown that fatty acid membranes could have served as stepping stones. They could have gradually incorporated more complex lipids, becoming more stable, more selective, more capable over time. Evolution doesn't require perfection from the start. It just requires something that works well enough to persist and improve. One of the most elegant things about these early membranes is how they could have facilitated their own reproduction. Imagine a vesicle containing RNA molecules that could replicate. As the RNA makes copies of itself, it uses up building blocks from inside the vesicle. This creates an osmotic imbalance. There are now more molecules inside than outside, creating pressure. The vesicle swells, taking in water. This stretches the membrane, creating gaps where new fatty acids from the environment can insert themselves. The membrane grows. Meanwhile, the RNA inside is producing something else, maybe short protein chains that help stabilize the membrane, or molecules that help capture more fatty acids from the environment. The vesicle that can do this better will grow faster, will be more stable, will be more likely to successfully divide into daughter vesicles that inherit both the RNA and the membrane. Natural selection has begun, not on organisms, but on chemical systems. But membranes presented a problem too. If they were too good at keeping things out, how could nutrients get in? How could waste products get out? Modern cells have elaborate protein channels and pumps to manage this. But early protocells had none of that machinery. They needed membranes that were selectively permeable, barriers that could discriminate without complex mechanisms. Temperature might have been key. Fatty acid membranes have a peculiar property. They become more permeable when heated. Near hydrothermal vents, where temperatures fluctuated, membranes might have cycled between open and closed states. Hot water from the vent makes them permeable, allowing materials to enter. Cooler water makes them less permeable, trapping materials inside. It's like a thermal pump, driven by environmental cycles, rather than biological machinery. The composition of the membrane mattered too. Pure fatty acid membranes are relatively permeable, but add in some other molecules, maybe some amino acids, or short peptides, or even mineral particles, and the properties change. Some additions make the membrane more stable, others make it more selective. This is starting to look like metabolism, isn't it? The controlled movement of materials across a boundary, the use of energy, in this case, thermal energy, to drive that movement, the production of molecules that maintain and improve the system. It's not the complex metabolism of modern cells, but it's a beginning. And there's something else about membranes that's crucial. They provide a surface. Chemical reactions happen faster on surfaces than in solution. Molecules stick temporarily, increasing their local concentration. They're oriented in particular ways, making certain reactions more likely. A membrane isn't just a barrier, it's a catalyst, an organizing principle, a scaffold for complex chemistry. Some scientists think membranes might have come first, before replication, before metabolism in any real sense. The lipid world hypothesis suggests that the ability to form compartments was the crucial innovation, and everything else followed. Compartments could concentrate random chemical reactions. Some combinations would be more successful than others, Over time, the most successful chemical networks would be those that could maintain and reproduce their compartments. But whether membranes came first, or replication came first, or they arose together, the crucial point is this: at some stage, they had to work together. A replicating molecule without a container is just chemistry. A container without heritable information is just a bubble, but together, they become something more. They become a system that can evolve, that can improve, that can eventually become alive. The transition might have been gradual. Maybe early replicators were just occasional visitors to vesicles, sometimes getting trapped inside during division. Maybe early membranes were just temporary shelters, forming and dissolving around chemical reactions. But at some point, the partnership became obligate. The replicators needed the membranes for protection and concentration. The membranes needed the replicators to encode the information for useful molecules. They became a unit, a proto-organism, the first draft of cellular life. And once that partnership was established, evolution could really begin to work, not just on molecules, but on organisms. Natural selection could favor better membranes, better replicators, better coordination between the two. The stage was set for the explosion of complexity that would follow. Part 5. The first spark of metabolism. Imagine you're watching a campfire. The wood doesn't just burst into flame. It needs a spark, sustained heat, the right amount of oxygen, too little air, and it smothers, too much wind, and it blows out. But get the conditions right, and you have a controlled burn that can last for hours, consuming fuel, releasing energy, maintaining itself against the cold night. Metabolism is like that fire, except instead of wood and oxygen, it uses chemical reactions to sustain the controlled burn of life. In those early vesicles bouncing in the primordial waters, something similar was trying to happen. Random chemical reactions were occurring, some releasing energy, others consuming it, but without coordination, without control. It was just chemistry happening. For life to truly begin, these reactions needed to be linked, organized into cycles and chains that could sustain themselves, and crucially, power the work of staying alive. The problem is fundamental. Everything tends toward disorder. It's the second law of thermodynamics, and it's non-negotiable. Leave anything alone long enough, and it falls apart. Mountains erode, stars burn out, complex molecules break down into simpler ones. Fighting this tendency requires energy, constantly applied, carefully directed. Life is, at its core, a very sophisticated way of postponing disorder. In modern cells, metabolism is breathtakingly complex. Hundreds of chemical reactions, precisely coordinated, with products from one reaction becoming reactants for another. Enzyme speed reactions, by factors of millions. Elaborate feedback loops maintain balance. It's like a city-sized chemical factory compressed into something smaller than a grain of sand. But the first metabolism couldn't have been anything like that. It had to be simple, robust, and capable of running without sophisticated machinery. One possibility is that metabolism began with surfaces, not membranes this time, but mineral surfaces. Iron-sulfur minerals, common near hydrothermal vents, have catalytic properties. They can facilitate electron transfers, speed certain reactions, provide a scaffold for organic molecules to interact. Some scientists think life might have begun as a kind of chemical coating on mineral surfaces, a metabolism-first scenario where self-sustaining reaction networks preceded both membranes and replication. Picture streams of warm, mineral-rich water flowing over porous rock near a hydrothermal vent. Organic molecules from the water stick to the rock surface. The minerals catalyze reactions between them. Some reactions produce molecules that stick even better, or that help catalyze other useful reactions. Over time, the surface becomes coated with an increasingly complex network of chemical processes, each supporting the others. It's not alive, but it's a kind of proto-metabolism, a self-sustaining chemical ecosystem. The acetyl-CoA pathway, also called the Wood-Lindahl pathway, might be a living fossil from these earliest times. It's one of the retry-key-continue-idit most ancient metabolic pathways we know, found in organisms that live in extreme environments similar to early Earth. It takes carbon dioxide and hydrogen and combines them to form acetyl-CoA, a molecule that's central to metabolism in all living things. The reactions can be catalyzed by iron-sulfur minerals. No complex enzymes needed, at least for the basic chemistry. This is profound. It means that the core of metabolism, the chemical heart that powers all life, might have started as simple chemistry on rock surfaces. The same reactions that modern cells perform with elaborate protein machinery might have originally happened spontaneously, slowly, crudely, on minerals at the bottom of ancient seas. But metabolism isn't just about running reactions, it's about coupling them, using energy from one reaction to drive another that wouldn't happen on its own. This is what ATP does in modern cells. It's the universal energy currency, the molecule that stores energy from food or sunlight and delivers it wherever it's needed. But ATP is complex, unlikely to have been present at life's beginning. So what came before? Maybe it was proton gradients. This sounds technical, but it's actually beautifully simple. When you have different concentrations of protons, hydrogen ions on two sides of a barrier, you have stored energy. It's like water behind a dam. Let the protons flow from high concentration to low, and you can use that flow to do work. Modern cells still use this principle. It's how mitochondria generate most of our ATP. Near hydrothermal vents, natural proton gradients exist. The vent fluid is alkaline, the ocean water more acidic. Where they meet, especially in the porous rock structures around vents, you have countless tiny chambers with proton gradients across their walls. These gradients represent free energy, available to any chemical system that could harness it. It's like nature providing free batteries, waiting for something to evolve that could use them. The first protocells might have been parasites on these natural gradients. They didn't generate their own energy, they simply positioned themselves where energy was freely available. Like the first humans who discovered fire by finding naturally occurring flames, then learned to maintain them before finally learning to create fire themselves. Evolution often works this way, opportunistically using what's available before developing the ability to create it independently. But there's another aspect to metabolism that's equally important: building things. Energy is useless without materials. Modern cells are masters of construction, taking simple molecules and assembling them into proteins, nucleic acids, lipids, carbohydrates, all the complex machinery of life. This biosynthesis had to start somewhere, and it probably started small. Simple amino acids, as we've discussed, form readily under prebiotic conditions, but linking them into proteins is harder. It requires removing water, which is thermodynamically unfavorable in water, obviously. Modern cells use sophisticated machinery and lots of ATP to force this to happen. But early protocells might have used simpler tricks. Wet-dry cycles, for instance. In a tide pool or near a hot spring, water evaporates, concentrating molecules and removing the water that prevents them from linking. When water returns, you have chains of amino acids, primitive proteins. They're random sequences, not encoded by genes, but some might have useful properties. Maybe they help stabilize membranes. Maybe they speed up certain reactions. Maybe they help capture energy from those proton gradients. The key is that metabolism didn't have to start perfect. It just had to start. A few reactions that could sustain themselves. A simple way to capture energy. Some basic ability to build necessary molecules. From that humble beginning, evolution could elaborate, optimize, and diversify. Each small improvement would be preserved if it helped the protocell survive and reproduce. And reproduction adds another dimension to metabolism. When a vesicle divides, it doesn't just need to copy its genetic material if it has any. It needs to ensure both daughter cells have functioning metabolism. This creates a strong selective pressure for robust self-organizing metabolic networks, ones that can spontaneously reassemble even when diluted or disrupted by division. This might explain why all life shares certain core metabolic pathways: the citric acid cycle, glycolysis, the pentose phosphate pathway. These aren't just similar across all domains of life, they're virtually identical. It's as if life found certain chemical solutions very early and never abandoned them. They became the foundation on which all subsequent evolution built. But here's a beautiful thought. Those early metabolic networks might not have been confined to individual cells. In the dense, interconnected world of early life, metabolic processes might have been shared. One protocell produces a waste product that another uses as food. Networks of different cell types, each specializing in different reactions, all dependent on each other. Life might have begun not as individual organisms, but as ecosystems, communities of interdependent chemical processes gradually becoming organized into discrete units. The transition from geochemistry to biochemistry might have been surprisingly smooth. The same types of reactions, just increasingly controlled and coordinated. The same energy sources, just captured more efficiently. The same building blocks, just assembled with greater precision. Life didn't invent new chemistry so much as it organized existing chemistry in new ways. This continuity is visible even today. The most ancient organisms on Earth, the archaea that live in extreme environments, have metabolisms that blur the line between biology and geology. They use hydrogen gas and carbon dioxide. They process sulfur and methane. They thrive at temperatures that would denature most proteins. Looking at them is like looking back in time, seeing echoes of how the first metabolism might have worked. Part 6. Information and Inheritance. Every living thing carries within it a message from the deep past, written in the 4-letter alphabet of DNA, edited and re-edited over billions of years, but still containing passages that date back to life's earliest chapters. This is the most extraordinary thing about life. It remembers, not consciously, but chemically. Every cell division, every generation, is an act of memory, passing forward information that has been accumulating since the very beginning. But DNA, elegant as it is, almost certainly wasn't the first genetic material. It's too complex, too specialized. It requires an elaborate support system of proteins to replicate and repair itself. It's like a library that needs trained librarians to function. The first genetic system had to be simpler, something that could work alone, or with minimal help. That something was probably RNA. RNA is DNA's older, more versatile cousin. Where DNA is stable, RNA is reactive. Where DNA just stores information, RNA can also perform chemical work. It's like the difference between a printed book and a person who has memorized the book and can also teach, translate, and improvise on its contents. In modern cells, RNA serves as the messenger between DNA and proteins, but increasing evidence suggests it once ran the whole show. In 1982, Thomas Cech discovered that some RNA molecules could catalyze reactions, just like protein enzymes. These ribozymes shattered the neat division between genetic material and chemical machinery. Here was a molecule that could be both the instruction manual and the worker following the instructions. It could, in principle, replicate itself without any help from proteins. This discovery launched the RNA world hypothesis, the idea that before the current DNA-protein world, there was a simpler world where RNA did everything. It stored information, catalyzed reactions, perhaps even formed simple structures. It was a less efficient world, a slower world, but a world that could work with far less complexity than what we see today. Creating self-replicating RNA in the laboratory has proven challenging but not impossible. In 2009, scientists created an RNA enzyme that could synthesize copies of other RNA molecules, including copies of itself. It was slow and error-prone, but it worked. More recently, researchers have created RNA systems that can replicate and evolve, growing more efficient over generations. These experiments suggest that an RNA world wasn't just possible, it was perhaps inevitable given the right conditions. But there's a problem with RNA. It's fragile. Heat breaks it apart. Alkaline conditions destroy it. Even in water, it slowly degrades. For RNA-based life to persist, it needed protection. This brings us back to those fatty acid vesicles, those protocells we discussed earlier. RNA inside a vesicle would be shielded from the worst environmental insults. The vesicle that happened to contain self-replicating RNA would have something no other vesicle had: heredity. Think about what heredity really means. It's the ability to pass traits from parent to offspring. Without it, every generation starts from scratch. With it, innovations can accumulate. A useful mutation doesn't die with the individual. It spreads through populations, becomes refined, combines with other useful mutations. Heredity is what allows evolution to climb mountains rather than randomly wandering the valleys. The first heredity was probably terrible by modern standards. Early RNA replicators might have had error rates of 10% or higher, one mistake for every 10 letters copied. Compare that to modern DNA replication, which makes about one error per billion letters. But high error rates aren't entirely bad when you're exploring possibilities. They generate diversity, create new variants to test against environmental challenges. In the early RNA world, sloppiness might have been a feature, not a bug. These errors, mutations, are evolution's raw material. Most would have been harmful, creating RNA molecules that couldn't replicate or that fell apart immediately. But occasionally, by pure chance, an error would improve something. Maybe the RNA would fold into a shape that made it more stable. Maybe it would replicate a bit faster. Maybe it would bind to the vesicle membrane in a way that prevented it from being lost during division. Here's where it gets interesting. RNA doesn't just encode information linearly like text on a page. It folds into complex three-dimensional shapes, and those shapes determine what it can do. A single mutation might drastically change the shape, and therefore the function. This means RNA can explore functional space very quickly, testing new capabilities with each copying error. Some of these capabilities might have been surprisingly sophisticated. Modern ribosomes can cut and splice RNA, link amino acids into proteins, even synthesize nucleotides. The ribosome, the massive molecular machine that builds all proteins in all living cells, is at its core a ribosome. It's arguably the most important molecule in biology, and it's living proof that RNA can perform complex chemical tasks. But RNA alone wasn't enough. Proteins, even short random ones, are better catalysts for most reactions. They're more stable, more versatile, can work in conditions that would destroy RNA. The first proteins were probably just short chains of amino acids, assembled randomly or by simple RNA templates. They wouldn't have been good for much, but even marginal help would have been selected for. The genetic code, the rules for translating RNA sequences into protein sequences, is nearly universal across all life. With minor variations, the same three-letter RNA sequences code for the same amino acids in bacteria, archaea, and eukaryotes. This suggests the code was established very early and has been frozen in place ever since. Changing it would be like trying to change the alphabet while writing, possible in principle, catastrophic in practice. But how did the code originate? It's not obvious why particular RNA triplets should correspond to particular amino acids. One fascinating hypothesis involves chemical affinity. Maybe certain amino acids naturally stick to certain RNA sequences. Over time, this weak chemical preference could have been formalized into a translation system. The code might not be arbitrary, but rather a frozen accident that began with simple chemistry. The transition from RNA to DNA probably came later, after basic metabolism and protein synthesis were established. DNA is more stable than RNA, better for long-term information storage, but it requires a complex enzyme to replicate, and that enzyme is made of protein. This creates a chicken and egg problem. DNA needs proteins to replicate, but proteins need DNA to store their blueprints. RNA, which can do both jobs poorly, might have been the bridge between no genetics and modern genetics. This transition wouldn't have happened overnight. For a long time, RNA and DNA might have coexisted, each with different roles. RNA remained the active messenger and occasional enzyme. DNA became the stable archive. Proteins took over most catalytic duties. This division of labor, DNA for storage, RNA for messaging, proteins for work, is so fundamental that it's found in every living cell. But information isn't just about storage and transmission. It's also about regulation, knowing when to use which information. Modern cells can turn genes on and off in response to environmental conditions. This requires sensory mechanisms, signal processing, decision-making of a sort. Even the simplest bacteria have remarkably sophisticated regulatory networks. The first regulatory systems were probably much simpler. Maybe certain RNA structures were stable only at certain temperatures. Maybe some proteins functioned only in the presence of specific metals. These conditional behaviors would allow protocells to respond to their environment in crude ways. Over time, these responses could become more sophisticated, more coordinated, more lifelike. We're approaching something that looks unmistakably alive now. Self-contained vesicles with semi-permeable membranes, metabolic networks capturing energy and building materials, genetic systems storing and transmitting information, regulatory mechanisms responding to environmental changes, each component supporting the others, the whole system capable of growth, division, and evolution. But there's still something missing from this picture. Life doesn't exist in isolation. It exists in communities, ecosystems, webs of interaction. The first cells weren't alone in the ocean. They were surrounded by other cells, other chemical experiments, other attempts at life, and the interactions between them would prove just as important as what happened inside them. Part 7. The Dance of Early Communities. Life never existed in isolation. From the very beginning, even before what we would properly call life, chemical systems influenced each other. Protocells competed for the same resources. They poisoned each other with waste products. They sometimes merged, exchanging contents, and occasionally, remarkably, they helped each other survive. The story of early life isn't just about individual cells figuring out how to replicate and metabolize. It's about communities learning to exist together, creating the first ecosystems in Earth's warm primordial waters. Imagine a small bay or lagoon on early Earth, protected from the worst ocean turbulence, warmed by volcanic heat, rich with minerals seeping from the rocks. This wasn't a sterile laboratory where one type of protocell could evolve in isolation. It was a crowded, chaotic experiment with millions of different chemical systems trying to persist simultaneously. Some wrapped in fatty acid membranes, others perhaps just organized films on mineral surfaces, some with RNA, others with different genetic polymers that didn't survive to the present, each one a different experiment in being. Competition was inevitable. They all needed the same basic resources: organic molecules for building, energy sources for driving reactions, space to exist. When resources are limited, which they always are, those who acquire them most efficiently survive. Those who don't, disappear. This is natural selection at its most basic, operating before what we would even call life, on chemical systems whose only crime was being less efficient than their neighbors. But competition alone doesn't explain what happened next. The pure competition should drive towards simplicity, the fastest replicator taking over everything. Instead, life became increasingly complex, diverse, interconnected. This happened because of a profound truth about chemistry and life. Waste from one process can be food for another. Consider a protocell that consumed amino acids to build primitive proteins. The process would produce waste, maybe ammonia, maybe other organic fragments. To the protocell producing them, these were useless or even toxic byproducts. But to another protocell with different chemistry, they might be valuable resources. A community begins to form, not through cooperation in any conscious sense, but through complementary chemistry. One cell's trash becomes another's treasure. These early metabolic exchanges would have been crude and inefficient, nothing like the precise nutrient cycles we see in modern ecosystems, but they established a principle that would shape all subsequent evolution. Life is interconnected. No organism is an island. Everyone's chemistry affects everyone else's. The waste products you generate, the resources you consume, the very fact of your existence changes the environment for everything around you. Horizontal gene transfer would have been rampant in these early communities. Modern bacteria can share genes through various mechanisms. They pick up naked DNA from the environment. They exchange plasmids through physical connections. They're infected by viruses that carry genes between cells. But early protocells, with their leaky membranes and simple organization, would have been even more promiscuous. Useful innovations, a better enzyme, a more stable membrane component, could spread through a population not just through inheritance, but through direct transfer. This creates a different picture of early evolution. Instead of a tree with distinct branches, imagine a web, with genetic information flowing in all directions. A useful gene arising in one lineage could quickly spread to others. Evolution wasn't just about competition between distinct species, but about the collective exploration of possibility by a community of interchanging parts. Viruses, or something like them, probably emerged very early in this process. As soon as there were cells with genetic material, there would have been parasitic genetic elements trying to hijack the machinery for their own replication. We tend to think of viruses as purely destructive, but they're also vectors for genetic exchange. They carry genes between hosts, sometimes integrating useful new capabilities. Many important innovations in the history of life might have spread first as viral infections before becoming permanent parts of the genome. The physical structure of these early communities mattered too. Protocells wouldn't have been evenly distributed through the water like a well-mixed solution. They would have accumulated on surfaces, in crevices, in zones where conditions were favorable. These accumulations, biofilms, we'd call them today, create their own microenvironments. The cells on the outside experience different conditions than those buried deeper. Chemical gradients form, specialization becomes possible, even advantageous. In a dense community, being identical to your neighbors is a disadvantage. You're competing for exactly the same resources. But if you can exploit a slightly different niche, maybe you're better at using a waste product others ignore. Maybe you can survive at slightly higher temperatures. Maybe you can attach to surfaces more firmly. You avoid direct competition. Diversity becomes a survival strategy. This might explain one of the most puzzling aspects of early evolution, the rapid diversification of metabolic strategies. Modern organisms use an astounding variety of energy sources. Some use sunlight. Others use hydrogen gas or sulfur compounds, or methane, or even electricity from rocks. This metabolic diversity probably arose very early, driven by the need to avoid competition in crowded communities. Communication, in a primitive sense, would have emerged in these communities, not language or even signaling as we typically think of it, but simple chemical influences. A protocell under stress might release molecules that affect its neighbor's behavior. Maybe these molecules make membranes more permeable, allowing faster nutrient exchange. Maybe they trigger dormancy-like states when resources are scarce. These wouldn't be intentional signals, just chemical consequences that happen to coordinate community behavior in useful ways. Cooperation too would have emerged, though not through any kind of planning or altruism. Simple physical proximity can create cooperation. If two protocells often end up near each other, maybe they stick to the same surfaces, maybe they're carried by the same currents, and they happen to have complementary metabolisms. They'll both do better when they're together. Over time, mechanisms that maintain this proximity would be selected for, the beginning of symbiosis. Some of these early symbioses might have been so successful that the partners became inseparable. This is the idea behind endosymbiosis. One cell living inside another, eventually becoming a permanent part of it. This definitely happened at least once, when bacterial cells took up residence inside archaea to become mitochondria, the powerhouses of eukaryotic cells. But simpler versions might have been happening from very early on, cells merging and separating, testing combinations, occasionally finding partnerships that worked. The environment shaped these communities in profound ways. Near hydrothermal vents, with their extreme temperature gradients and chemical richness, communities would be dense but stressed. In calmer waters, with fewer resources but more stability, different strategies would succeed. Tidal zones, with their cycles of wet and dry, would favor organisms that could survive dramatic changes. Each environment would create its own unique community, its own solutions to the challenge of staying alive. Climate events would have periodically disrupted these communities. Volcanic eruptions could poison entire regions with sulfur compounds. Asteroid impacts could vaporize seas. Ice ages, yes, they happened even on early Earth, could lock up water and concentrate salts to lethal levels. But disruption also creates opportunity. After each catastrophe, the survivors would rapidly diversify to fill empty niches. These boom and bust cycles would drive evolution faster than stable conditions ever could. Some communities might have modified their own environments in ways that created new opportunities. Maybe they produced molecules that precipitated minerals, creating physical structures that other cells could colonize. Maybe they generated gases that changed local pH, creating gradients that could be exploited for energy. These early examples of ecosystem engineering would have created feedback loops, life shaping the environment which shapes life. The waste products of these communities would have begun to accumulate in the environment. Oxygen, which would later transform the planet, was initially just a toxic waste product of certain types of photosynthesis. Methane, produced by early archaea, would have been a greenhouse gas helping to warm the planet under the dim young sun. Life wasn't just adapting to the environment, it was beginning to change it on a global scale. Part 8. Learning to capture light. 4 billion years ago, the sun rose every 6 hours over a world that couldn't see it. No eyes existed to perceive its light, no leaves to capture its energy. The sun's rays struck bare rock and empty water, delivering vast amounts of energy that mostly just heated surfaces and drove weather. All that power streaming past a planet that hadn't yet learned how to catch it. But deep in the water, chemistry was about to discover the most important trick in the history of life: how to eat sunlight. The sun delivers more energy to Earth in 1 hour than all human civilization uses in a year. It's a nuclear furnace 93 million miles away, converting millions of tons of matter into energy every second. And Earth intercepts just a tiny fraction of that output. But that tiny fraction is enormous by biological standards. Learning to tap into it would transform life from a marginal chemical curiosity scraping by on geological energy into a planetary force capable of reshaping the atmosphere, the climate, and the very rocks themselves. The first attempts to capture light probably weren't photosynthesis as we know it. They might have been simpler, cruder, molecules that absorbed light and used the energy to drive simple chemical reactions. Some modern bacteria have rhodopsins, light-sensitive proteins that can pump protons across membranes when struck by photons. It's not true photosynthesis, it doesn't fix carbon or produce oxygen, but it's a way of converting light into usable energy. Something similar might have been life's first solar technology. But the real revolution came with chlorophyll, or something like it. Chlorophyll is a remarkable molecule, a ring of carbon and nitrogen with a magnesium atom at its heart. When light strikes it, an electron gets excited, jumps to a higher energy level. That excited electron can be passed to other molecules. It's extra energy used to drive chemical reactions. It's like a molecular solar panel, converting photons into electrical current at the atomic scale. The first photosynthetic organisms probably didn't produce oxygen. They likely used hydrogen sulfide or other compounds as electron donors, producing sulfur as waste. We still see this ancient form of photosynthesis in purple and green sulfur bacteria, living fossils from an earlier era. They cluster around volcanic vents and in oxygen-poor waters, practicing a form of photosynthesis that predates the oxygen revolution by perhaps 1 billion years. These early photosynthesizers would have had a huge advantage. While other proto-cells competed for limited chemical resources, they could tap directly into the sun's endless supply. They weren't dependent on volcanic heat or chemical gradients. Anywhere sunlight could reach, they could survive. This opened up vast new territories. The surface waters of the entire ocean became potential habitat. But light, especially ultraviolet light, is also dangerous. It breaks chemical bonds, damages genetic material, tears apart complex molecules. Early photosynthesizers had to balance the benefit of energy capture against the cost of radiation damage. They probably started deep enough in the water to be protected from the worst UV, moving toward the surface only as they evolved better repair mechanisms and protective pigments. The evolution of oxygenic photosynthesis, the kind that produces oxygen as waste, was perhaps the most consequential innovation in the history of life. It probably arose only once, in the ancestors of cyanobacteria, somewhere around 2.5 to 3 billion years ago. The exact timing is debated, written ambiguously in rocks that preserve chemical hints but not certainty. Oxygenic photosynthesis is more complex than the older forms. It requires two photosystems working in concert, one to split water molecules and another to fix carbon dioxide. It's like having two solar panels wired in series, each capturing different wavelengths of light. The complexity suggests it didn't arise all at once, but was assembled over time from simpler components. The key innovation was learning to split water. Water is everywhere, unlike hydrogen sulfide or other electron donors that early photosynthesis relied on. But water is also very stable. Breaking it apart requires significant energy. The photosynthetic machinery that could do this, that could literally tear water molecules apart using sunlight, was biochemical dynamite. It gave access to an essentially unlimited electron source. But this innovation came with a terrible price, or what seemed terrible to most life at the time: oxygen waste. Oxygen is highly reactive. It steals electrons from other molecules, breaking them apart. To the anaerobic life that dominated early Earth, oxygen was poison. As cyanobacteria spread, and oxygen began accumulating, first in local pockets, then regionally, then globally, it created the first great pollution crisis in Earth's history. This wasn't immediate. For hundreds of millions of years, oxygen was absorbed as fast as it was produced. It reacted with dissolved iron in the oceans, creating banded iron formations we can still see in rocks today, layers of rust, essentially, marking the slow oxidation of an entire planet. It reacted with methane in the atmosphere, with sulfur compounds, with organic matter. Earth had a lot of chemical reducing power that had to be exhausted before oxygen could accumulate freely. But eventually, perhaps around 2.4 billion years ago, free oxygen began building up in the atmosphere. The Great Oxidation Event it's called, though it was more of a great poisoning from the perspective of most life at the time. Anaerobic organisms retreated to oxygen-free refuges, deep sediments, stagnant waters, anywhere the terrible gas couldn't reach. Many lineages went extinct. It was arguably the greatest catastrophe life had ever faced, and life itself was the cause. Yet from crisis came opportunity. Oxygen, for all its dangers, is also rocket fuel for metabolism. Organisms that could tolerate it, then use it, had access to far more energy than anaerobic metabolism could provide. Aerobic respiration yields about 18 times more energy from the same food compared to fermentation. It's the difference between a campfire and an explosion. The cyanobacteria themselves had to evolve protections against their own waste product. Antioxidant molecules, enzymes that could neutralize reactive oxygen species, compartmentalization, to keep oxygen-generating processes separate from oxygen-sensitive ones. They became chemical warfare specialists, wielding a weapon that killed competitors while developing armor against their own toxin. As oxygen accumulated, it began to change the planet in ways beyond the biological. In the upper atmosphere, oxygen molecules were split by UV radiation and recombined as ozone. The ozone layer began forming, a shield that would eventually allow life to colonize land by blocking the most damaging radiation. The sky itself changed color, from the orange-pink of a methane-rich atmosphere to the blue we know today. Photosynthesis also began to draw down carbon dioxide from the atmosphere. CO2 had been the dominant greenhouse gas, keeping Earth warm under the dim young sun. As photosynthesizers pulled it from the air and locked it into organic matter, some of which was buried in sediments, the planet cooled. Too much, it turns out. The drawdown of greenhouse gases combined with the destruction of methane by oxygen triggered a series of ice ages, including potentially a snowball Earth event where ice covered the planet from poles to equator. But life persisted, adapted, diversified. In the oxygen-rich waters, new forms of photosynthesis evolved. Red algae, green algae, each with slightly different pigments capturing different wavelengths of light. Competition for light drove diversification in light harvesting strategies. Some organisms developed pigments that could capture the dim blue light that penetrates deepest into water. Others specialized in the green light that other photosynthesizers reflected. Photosynthesis became more efficient over time. Antenna complexes evolved, arrays of pigment molecules that could funnel captured light energy to reaction centers. Mechanisms for concentrating CO2 improved, allowing photosynthesis to continue even as atmospheric CO2 levels dropped. The C4 antenna photosynthesis pathways, though they wouldn't evolve for billions of years, would eventually allow plants to photosynthesize efficiently in hot, dry conditions. The impact of photosynthesis extended beyond just producing oxygen and fixing carbon. Photosynthetic organisms became the base of food webs, the primary producers that all other life would ultimately depend on. They created gradients, oxygen-rich surface waters and oxygen-poor depths that other organisms could exploit. They produced compounds that other organisms would learn to use or break down, driving the evolution of new metabolic pathways. Photosynthesis also created the possibility of complex multicellular life. The energy available from aerobic respiration, breathing the oxygen that photosynthesis produced, would eventually power the evolution of animals, fungi, and complex plants. Every breath you take is possible because billions of years ago, some bacteria learned to split water with sunlight. Part 9. The Architecture of Cooperation. 3 billion years ago, life faced a problem. Individual cells had become about as sophisticated as they could be, while remaining single. They had evolved elaborate metabolisms, complex genetic regulatory systems, sophisticated membranes studded with protein channels and pumps. But there's only so much you can fit into a microscopic sphere. To become more complex, life needed a new strategy. It needed to learn how to cooperate at a scale never before attempted. It needed to invent multicellularity. But before true multicellularity, there were experiments in togetherness, cells that stuck together after division instead of drifting apart, colonies where individuals remained distinct but gained advantages from proximity, biofilms, where different species created structured communities. These weren't just random aggregations, they were the first attempts at something profound, the organization of life above the cellular level. Consider a simple question: why should cells cooperate? From a selfish gene perspective, each cell should focus on its own replication. Helping neighbors who might be competitors seems counterproductive. But mathematics and chemistry conspire to make cooperation advantageous under certain conditions. When cells are related, daughters of the same parent, helping each other can be like helping copies of your own genes. When cells have complementary abilities, working together can achieve things neither could alone. The simplest form of cooperation is just sticking together. Many bacteria produce extracellular polymers, essentially slime, that helps them adhere to surfaces and to each other. This isn't sophisticated cooperation, but it has advantages. A group of cells can resist being washed away by currents better than individual cells. They can create local chemical environments different from the surrounding medium. They can share nutrients and signaling molecules more efficiently. Modern stromatolites give us a window into how early cooperation might have looked. These layered structures, built by communities of cyanobacteria and other microbes, have been forming in shallow waters for over 3.5 billion years. They're not organisms, they're more like apartment buildings created by microscopic life. Each layer represents a generation of cells that lived, photosynthesized, and trapped sediment, creating a platform for the next generation to build upon. In these structures, we see the beginning of differentiation. Cells at the surface, exposed to light, photosynthesize actively. Cells deeper in the structure, where light doesn't penetrate, switch to different metabolisms. Some specialize in nitrogen fixation, others break down waste products. It's not true multicellularity. The cells could survive independently, but it's a community where different positions mean different roles. Chemical communication would have been crucial for these early communities. Quorum sensing, where cells detect the concentration of signaling molecules to determine population density, probably evolved very early. When enough cells are present, when the signal reaches a threshold, behaviors change. Genes turn on or off. Individual cells begin acting as parts of a collective. This kind of coordination can be remarkably sophisticated, even in simple organisms. Modern bacteria using quorum sensing can coordinate attacks on host organisms, waiting until their numbers are sufficient to overwhelm immune defenses. They can synchronize the production of light, creating the eerie glow of bioluminescent bays. They can build complex structures, timing the production of materials so that architectural elements appear in the right order. But the transition from colonies to true multicellularity required something more. Cells had to give up their independence. In a true multicellular organism, most cells never reproduce independently. They're born, they perform their function, and they die without ever passing on their genes directly. Only specialized reproductive cells get that privilege. From an evolutionary perspective, this seems like a terrible deal for the non-reproductive cells. The solution to this paradox lies in relatedness. In a multicellular organism developing from a single fertilized cell, all cells are genetically identical, barring mutations. A muscle cell helping a sperm or egg cell reproduce is helping an exact copy of its own genome. It's like worker bees, sterile themselves, helping their queen sister reproduce. The genes get passed on even if the individual doesn't reproduce directly. This genetic unity allows for extreme cooperation. Cells can specialize to degrees impossible when they need to maintain all capabilities for independent survival. Some cells become pure energy generators. Others become structural support. Some become chemical factories producing compounds for the entire organism. Some even become programmed to die at specific times, sacrificing themselves for the good of the whole. The evolution of programmed cell death, apoptosis, was crucial for complex multicellularity. It seems paradoxical, genes that cause their carriers to die should be selected against. But in a multicellular context, controlled cell death is essential. It sculpts developing tissues, removes damaged cells, creates spaces for other structures. Your fingers exist as separate digits because cells between them died during development. Every second, millions of your cells are killing themselves to maintain your body's organization. Communication systems became more sophisticated in multicellular organisms, not just chemical signals diffusing between cells, but direct physical connections. gap junctions that allow small molecules to pass directly between adjacent cells, plasmodes mater in plants that create continuous cytoplasmic connections. These connections turn individual cells into something more like a network, a distributed biological computer where information and resources flow freely. The extracellular matrix evolved as another form of cooperation. Cells began secreting not just slime but structured proteins, collagen, elastin, fibronectin. These created scaffolds that cells could build upon, highways they could migrate along, repositories for growth factors and signals. The matrix became as important as the cells themselves, an external skeleton that gave multicellular organisms their shape and strength. But multicellularity created new problems. How do you coordinate billions or trillions of cells? How do you ensure that cells in your liver behave like liver cells and not like neurons? How do you prevent cheetah cells that consume resources without contributing? These challenges drove the evolution of increasingly sophisticated regulatory systems. Development became a carefully choreographed process. Chemical gradients established body axes, which end becomes the head, which the tail. Waves of gene expression created segments and layers. Master regulatory genes controlled entire cascades of downstream genes, turning on developmental programs that could build eyes, limbs, or organs. The same basic toolkit, modified and redeployed, could create the vast diversity of multicellular forms. Cancer represents the dark side of multicellularity. What happens when cells stop cooperating? A cell accumulates mutations that let it ignore growth signals, evade death signals, replicate indefinitely. It becomes a cheetah. Reveratory key continued itting to unicellular selfishness within a multicellular body. The fact that cancer exists in virtually all multicellular organisms shows how difficult it is to maintain cooperation among trillions of cells. Evolution has created elaborate tumor suppression mechanisms, redundant checkpoints, immune surveillance, and still, given enough time, cheetahs arise. The immune system itself represents a sophisticated form of cellular cooperation. Specialized cells patrol the body, identifying and eliminating threats, not just foreign invaders, but also misbehaving native cells. They communicate through complex molecular languages, remember past threats, coordinate responses involving millions of cells. It's a distributed defense network that requires extraordinary cooperation to function. But perhaps the most remarkable form of cellular cooperation emerged in nervous systems. Neurons gave up the ability to divide in exchange for extreme specialization in information processing. They developed elaborate branches for receiving and transmitting signals. They formed networks of stunning complexity. Your brain contains roughly 86 billion neurons making perhaps 100 trillion connections. These cells cooperate to create something unprecedented: consciousness, memory, the ability to model the world and imagine futures. The evolution of multicellularity wasn't a one-time event. It happened independently at least 25 times, in animals, plants, fungi, various algae, slime molds, and several bacterial lineages. Each time, different solutions were found to the same basic challenges. Plants evolved rigid cell walls and plasmode smata. Animals evolved flexible cells and complex signaling. Fungi evolved multicellular filaments that can act as individuals or fuse into networks. The diversity of solutions shows how powerful the advantages of multicellularity must be. Size was one obvious advantage. Multicellular organisms could grow large enough to access new resources, resist predation, create internal environments independent of external conditions. But size brought its own challenges. How do you move nutrients to cells far from the surface? How do you remove waste? How do you coordinate activities across distances that from a cellular perspective might as well be different continents? Transport systems evolved, circulatory systems in animals, vascular tissues in plants. These weren't just pipes but active networks that could regulate flow, respond to demand, repair damage. The human circulatory system, with its 60,000 miles of blood vessels, represents an engineering feat that distributes resources to 37 trillion cells, each no more than a few cell widths from a capillary. Multicellularity also enabled division of labor at a grand scale. In a colony of identical cells, everyone has to be a generalist. But in a multicellular organism, cells could become extreme specialists. Photoreceptor cells that do nothing but detect light. muscle cells packed with contractile proteins, neurons that gave up most metabolic functions to maximize information processing. This specialization allowed multicellular organisms to be far more efficient than colonies of generalists. The fossil record shows that complex multicellularity, organisms with differentiated tissues, arose sometime before 600 million years ago. The Ediacaran fauna, strange quilted and frondose organisms unlike anything alive today, represent early experiments in large body size and complex organization. Most of these experiments failed, leaving no modern descendants, but some succeeded spectacularly. The Cambrian explosion, around 540 million years ago, saw the rapid appearance of most major animal body plans: arthropods with jointed legs and compound eyes, mollusks with shells and radulas, chordates with notochords and neural tubes. This wasn't the origin of multicellularity. That happened much earlier, but it was when multicellular organisms discovered how to build complex mobile bodies with specialized organs. Plants conquered land around 450 million years ago, bringing multicellularity to a new frontier. They had to evolve waxy cuticles to prevent desiccation, stomata to regulate gas exchange, rigid tissues to stand against gravity. They created forests that transformed terrestrial landscapes, soil that could support complex ecosystems, and ultimately the oxygen-rich atmosphere that large animals require. Each innovation in multicellular organization opened new possibilities. The evolution of flowers created partnerships with insect pollinators. The evolution of seeds allowed plants to package embryos with food supplies. The evolution of fruit turned animals into seed dispersers. Multicellularity didn't just create complex organisms, it created complex ecological relationships. Part 10. The Oxygen Revolution. 2.5 billion years ago, Earth experienced the most dramatic pollution crisis in its history. The pollutant wasn't toxic waste from some industrial process, it was oxygen, produced by life itself. What we breathe without thinking, what we consider essential for existence, was once a deadly poison that nearly ended life's experiment entirely. The Great Oxidation Event wasn't just a change in atmospheric chemistry, it was a revolution that transformed the planet's surface, its climate, and the very nature of life itself. For the first 1.5 billion years of life's existence, Earth was an anaerobic world. The atmosphere contained virtually no free oxygen. Methane and ammonia were common gases. Iron dissolved readily in the oceans. Sulfur chemistry dominated many biological processes. Life had evolved exquisite adaptations to this oxygen-free world. Enzymes that would be poisoned by oxygen, metabolic pathways that required its absence, DNA repair mechanisms that didn't need to handle oxidative damage. Then cyanobacteria changed everything. These photosynthetic bacteria had evolved something unprecedented: the ability to split water molecules using sunlight, extracting electrons and dumping oxygen as waste. It was like discovering nuclear power, tremendously powerful but incredibly dangerous. The oxygen they produced was reactive, corrosive, deadly to most existing life forms, but it was also abundant. Water was everywhere, unlike the limited supplies of hydrogen sulfide or iron that earlier photosynthesizers had relied on. At first, the oxygen was absorbed as quickly as it was produced. The oceans were full of dissolved iron, and oxygen readily combined with it to form iron oxides, rust. These precipitated out, creating the banded iron formations we see in rocks from this period. Layer after layer of rust, recording millions of years of oxygen production that never made it to the atmosphere. The oceans were literally rusting. Methane in the atmosphere also consumed oxygen, combining to form carbon dioxide and water. Reduced sulfur compounds were oxidized. Organic matter was burned. Earth had an enormous oxygen debt. Chemicals that would readily react with oxygen, but had to be paid before free oxygen could accumulate. It was like trying to fill a bucket with countless holes. For perhaps hundreds of millions of years, cyanobacteria pumped out oxygen that immediately disappeared into chemical reactions. But eventually, gradually, the sinks began to fill. The dissolved iron was exhausted. The atmospheric methane was depleted. Somewhere around 2.4 billion years ago, the exact timing is still debated, free oxygen began accumulating in the atmosphere. Not much at first, perhaps 1 to 2% of today's levels, but enough to fundamentally change the planet's chemistry. For anaerobic life, this was catastrophic. Oxygen is highly electronegative. It steals electrons from other molecules, breaking them apart. Proteins denature, lipids peroxidize, DNA suffers strand breaks. Many metabolic pathways simply stop working in oxygen's presence. Imagine if the air suddenly became filled with chlorine gas. That's what oxygen was like for organisms that had evolved in its absence. The anaerobes retreated to refugia, deep sediments where oxygen couldn't penetrate, stagnant waters where it was quickly consumed, the guts of other organisms where it was excluded. Many lineages went extinct entirely. Others survived but were marginalized, pushed to the edges of the biosphere. The rulers of the early Earth became refugees in their own world. But evolution doesn't waste opportunities. Some organisms evolved tolerance to oxygen, enzymes like catalase and superoxide dismutase that could neutralize reactive oxygen species before they caused damage. These were like hazmat suits, allowing cells to survive in the presence of the toxic gas, even if they couldn't use it. Then came the real innovation, aerobic respiration. Some cells learned not just to tolerate oxygen, but to use it. Oxygen's tendency to steal electrons, so destructive to unprepared cells, could be harnessed to extract energy from food far more efficiently than fermentation. Where fermentation might extract 2 units of ATP from a glucose molecule, aerobic respiration could extract 36. It was like upgrading from a wood stove to a jet engine. This efficiency advantage was overwhelming. Aerobes could grow faster, compete better, support more complex metabolisms. They began to dominate environments where oxygen was available. The toxic waste of cyanobacteria became rocket fuel for a new kind of life. The polluters and the adapted were often the same organisms. Cyanobacteria had to evolve protections against their own waste product. The oxygenation of the atmosphere had cascading effects. Methane, a powerful greenhouse gas that had helped keep early Earth warm despite the dim young sun, was destroyed by oxygen. The planet cooled dramatically, potentially triggering the Huronian glaciation, a series of ice ages that may have covered the entire planet in ice. Life had not just polluted its environment, it had disrupted the climate system. But oxygen also created new possibilities. In the upper atmosphere, oxygen molecules O2 were split by ultraviolet radiation, some recombined as ozone O3, creating a layer that absorbed UV radiation. This ozone layer would eventually make it possible for life to colonize land without being destroyed by radiation. The pollutant became a shield. The chemistry of the oceans changed profoundly. Elements that had been common in dissolved form, iron, manganese, uranium, became insoluble oxides and precipitated out. New minerals formed that had never existed on Earth before. The color of the oceans probably changed from green, from dissolved iron to blue. The smell certainly changed, from the rotten egg stench of hydrogen sulfide to something closer to what we know today. Oxygen also enabled new biological innovations. Collagen, the protein that gives animals structural strength, requires oxygen for its synthesis. Complex multicellularity, with its energy-intensive cellular differentiation and signaling, might not have been possible without the efficiency of aerobic metabolism. The oxygen revolution set the stage for the eventual emergence of complex life. But perhaps most importantly, oxygen created gradients, differences in oxygen concentration that organisms could exploit. The surface waters became oxygen-rich, while deeper waters remained oxygen-poor. Day-night cycles created temporal gradients, as photosynthesis produced oxygen during the day. These gradients represented opportunities for organisms that could move between different oxygen zones or tolerate varying concentrations. The sulfur cycle became more complex as oxygen enabled new transformations. Sulfate-reducing bacteria could use oxidized sulfur as an electron acceptor in the absence of oxygen. Sulfur-oxidizing bacteria could extract energy from reduced sulfur compounds in the presence of oxygen. Entire communities developed around sulfur cycling, creating some of the most productive ecosystems in oxygen minimum zones. Nitrogen cycling also intensified. The production of nitrate through nitrification required oxygen. The nitrification, converting nitrate back to nitrogen gas, occurred in oxygen-poor zones. The nitrogen cycle became a complex dance between oxic and anoxic processes, each dependent on the other, creating a global biogeochemical cycle that continues today. The geological record of the Great Oxidation Event is written in many forms. Red beds, Sedimentary rocks, colored by oxidized iron, appear for the first time. Uranium deposits show evidence of oxidative weathering. Sulfur isotopes show a shift indicating atmospheric oxygen. It's like a crime scene with multiple forms of evidence all pointing to the same culprit, life itself. The transition wasn't smooth or complete. For perhaps 1 billion years after the initial oxygenation, atmospheric oxygen levels remained relatively low and unstable. There were periods of increased oxygenation and periods where oxygen levels crashed. These fluctuations might have been caused by changes in productivity, weathering rates, or volcanic activity. Life and Earth were still negotiating their relationship. Around 800 million years ago, oxygen levels began rising again in what's called the Neoproterozoic oxygenation event. This second rise might have been triggered by the evolution of eukaryotic algae, more efficient at photosynthesis than cyanobacteria, or it might have been caused by increased burial of organic matter, preventing it from being oxidized and consuming oxygen. Whatever the cause, this second oxygenation coincided with the appearance of the first animals. Animals are oxygen addicts. Even the simplest animals require oxygen levels of at least 0.5-1% of present atmospheric levels. Complex animals with circulatory systems and active lifestyles need much more. The Cambrian explosion of animal diversity around 540 million years ago coincided with oxygen levels approaching modern values. Oxygen didn't just allow animals to exist, it powered their diversification. Part 11: Minerals and Life's Chemistry. Look at a rock, any rock. That solid, seemingly simple object contains a history written in minerals, crystals that formed under specific conditions of temperature, pressure, and chemistry. But here's something remarkable: about 2/3 of all mineral species on Earth exist only because life exists. Before life, Earth had perhaps 1,500 different minerals. Today we know of over 5,000. Life didn't just adapt to Earth's geology, it fundamentally transformed it, creating a planet whose very rocks tell the story of biology. In the beginning, Earth's minerals were simple. The planet inherited about 60 different minerals from the solar nebula, basic compounds like diamond, graphite, and some simple silicates that could form in the vacuum of space. As the planet differentiated, with heavy elements sinking and lighter ones rising, new minerals formed. Volcanic activity created more. Water, when it finally condensed, enabled solution chemistry that produced yet more minerals. By the time life appeared, Earth had developed perhaps 1,500 mineral species through purely geological processes. But life changed the game entirely, not immediately. The first cells were too few, too simple to affect planetary mineralogy. But as life spread and diversified, as it began producing oxygen and other reactive compounds, as it learned to precipitate shells and bones, it became a geological force. Life became Earth's most creative mineralogist. Consider what happens when oxygen enters the picture. Before the Great Oxidation Event, copper existed mainly as sulfides or native metal. After oxygenation, copper could form hundreds of different oxide, carbonate and sulfate minerals, blues and greens that had never existed on Earth before. Malachite, azurite, turquoise. These gorgeous minerals owe their existence to life's oxygen. Iron mineralogy was completely transformed. Before oxygen, iron moved freely through the environment in its reduced form. After oxygen, it formed countless oxide minerals: hematite, magnetite, goethite. The banded iron formations, those spectacular striped rocks from the time of the Great Oxidation Event, represent a mineralogical transition that could only happen once in a planet's history, when life first poisoned its world with oxygen. But oxygen was just the beginning. Life learned to precipitate minerals directly, using them for structure and protection. Cyanobacteria began precipitating calcium carbonate, creating stromatolites. Other organisms followed suit. Diatoms built intricate shells from silica. Foraminifera created tests from calcium carbonate. Radiolarians constructed elaborate skeletons from strontium sulfate. Each group of organisms became a biological mineral factory. The chemistry involved is sophisticated. Organisms don't just precipitate minerals randomly, they control the process with exquisite precision. Proteins template crystal growth, determining shape and size. Organic molecules are incorporated into crystal structures, changing their properties. The result is biominerals that are nothing like their geological counterparts. Stronger, tougher, more complex. Consider nacre, or mother of pearl. It's made of the same calcium carbonate found in chalk, but organized completely differently. Tiny aragonite tablets are glued together with organic polymers in a brick and mortar structure. The result is 3,000 times tougher than geological aragonite. It's a composite material that human engineers have only recently learned to mimic. Or magnetite in magnetactic bacteria. These organisms precipitate perfect nanocrystals of magnetite, each exactly the right size to be a single magnetic domain. They arrange these crystals in chains that act as compass needles, allowing the bacteria to navigate along Earth's magnetic field lines. It's biological nanotechnology that preceded human nanotechnology by billions of years. Life's influence on mineralogy extends beyond direct precipitation. Organic acids secreted by lichens and plant roots weather rocks in ways that purely chemical processes cannot. This weathering creates new minerals, clays that form only in soils, phosphates that concentrate in guano deposits, evaporite minerals modified by bacterial metabolism. The deep carbon cycle was transformed by life. Carbon that would have remained in the atmosphere as CO2 was pulled down by photosynthesis, incorporated into organic matter, sometimes buried in sediments. This organic matter, subjected to heat and pressure, became coal, oil, and natural gas. But it also participated in mineral formation, carbonate minerals that lock carbon away for millions of years. Some minerals form only in the presence of specific biological processes. Take the uranium mineral deposits in ancient rocks. Many formed when uranium, mobile in its oxidized state, encountered organic matter or bacterial communities that reduced it, causing it to precipitate. Without life creating reducing conditions in an oxidizing world, these deposits wouldn't exist. Clay minerals tell their own story of life's influence. While some clays form through purely inorganic processes, others are products of biological weathering. Soil clays often contain signatures of the organisms that helped create them. Isotopic ratios, trace elements, organic molecules trapped in their layered structures. They're like mineralogical fingerprints of ancient ecosystems. The phosphorus cycle was completely altered by life. Phosphorus, essential for DNA, RNA, and cellular energy, is often a limiting nutrient. Organisms evolved mechanisms to concentrate it, in bones, in teeth, in shells. When these organisms died, their phosphate-rich remains sometimes formed massive deposits. The phosphorate deposits we mine for fertilizer are essentially concentrated ancient life. Even cave minerals show life's hand. Many cave formations, stalactites, stalagmites, flowstones, form through purely geological processes, but others require biological mediation. Bacteria can precipitate minerals in caves, creating formations impossible without life. Some cave minerals form only when bat guano interacts with limestone, creating exotic phosphates and nitrates. Life also prevented certain minerals from forming. Before photosynthesis, minerals like pyrite, iron sulfide, formed readily in the oceans. After oxygenation, sulfide minerals became restricted to oxygen-poor environments. Life had created chemical conditions that excluded certain minerals from most of Earth's surface. The silicon cycle was revolutionized when organisms learned to precipitate silica. Diatoms, radiolarians, and glass sponges pull dissolved silicon from seawater, creating intricate structures. When they die, their silica skeletons accumulate, sometimes forming entire rock formations. Diatomaceous earth, radiolarate, spiculite. These rocks are essentially fossilized glass sculptures created by life. Billion mineralization became increasingly sophisticated over evolutionary time. Early organisms might have precipitated minerals accidentally or as waste products, but natural selection favored those who could control the process. Minerals became tools, teeth for grinding, shells for protection, bones for support, lenses for focusing light. The minerals in your own body tell this story. Your bones aren't just calcium phosphate. They're a complex composite with the mineral phase intimately associated with collagen proteins. Your teeth contain the hardest biological material known, enamel that's 96% mineral, but organized in ways geology never achieves. Even your inner ear contains tiny crystals, otoliths, that help you maintain balance. Some organisms became mineral extremists. Certain bacteria thrive in acid mine drainage, precipitating iron minerals in conditions that would dissolve most rocks. Others live in boiling springs, creating sulfur and arsenic minerals. Still others inhabit salt lakes, precipitating minerals so unusual they weren't known until life was found making them. The co-evolution of life and minerals continues today. As organisms evolve new metabolisms, they create conditions for new minerals to form. As new minerals appear, organisms evolve to exploit them. It's a dance that's been going on for billions of years, each partner changing the other, creating the extraordinary mineralogical diversity we see today. Human activity represents the latest chapter in this story. We've created entirely new minerals, synthetic compounds that never existed before. Some formed accidentally in mine tailings or industrial waste. Others were deliberately synthesized. We've become a geological force ourselves, following in the tradition of life as a transformer of planetary mineralogy. Part 12. The day and night cycle begins. 4 billion years ago, the sun rose and set every 6 hours. The early Earth spun like a frantic dancer, whirling through days so brief that life, had it existed then in any complex form, would have been dizzy with the speed of it all. But life didn't just adapt to Earth's rhythms, it changed them, and in turn those changing rhythms shaped life in ways we're only beginning to understand. The story of how days became days, our days, 24-hour days, is also the story of how life learned to tell time. The moon was the great break on Earth's rotation, hanging 10 times closer than it does today. Its gravitational pull created massive tides, not just in the nascent oceans, but in the solid rock itself. These tides dissipated energy, gradually slowing Earth's spin like friction slowing a spinning top. But this was a slow process, playing out over billions of years. Each day grew longer by perhaps 20 microseconds per year, imperceptible on human timescales, but transformative over geological time. For early life, these rapid days meant constant change. 6 hours of light, 6 hours of darkness, assuming you were at the surface to experience it at all. The temperature swings would have been dramatic, heating quickly under the young sun, cooling rapidly in the brief nights. Any organism trying to photosynthesize had only a few hours of useful light before darkness returned. Any process that required sustained conditions had to cope with this relentless cycling. But life is remarkably good at finding patterns and exploiting them. Even the simplest bacteria today have circadian rhythms, internal clocks that track the daily cycle. These aren't learned behaviors, but hard-coded genetic programs, molecular machinery that counts out roughly 24 hours, even in constant conditions. The ancestor of these clocks must have evolved very early, when days were much shorter, and then adjusted as the planet slowed. The first biological clocks were probably simple, perhaps just chemical reactions that ran at different rates in light and darkness. But natural selection favored organisms that could anticipate the cycle rather than just respond to it. If you could prepare for sunrise before it happened, start producing photosynthetic machinery in advance, you'd have an advantage over organisms that waited for the light to switch them on. As days lengthened from 6 hours to 8, to 10, to 12, life had to constantly recalibrate. This wasn't a smooth process. The moon's distance changed, continental positions shifted, even massive impacts could alter rotation rates. Life had to evolve flexibility in its timekeeping, the ability to adjust to changing day lengths while maintaining essential rhythms. Cyanobacteria, those oxygen-producing revolutionaries, became masters of time. They separated incompatible processes temporarily. Nitrogen fixation at night, when oxygen production ceased, since the nitrogen-fixing enzyme is poisoned by oxygen. DNA repair during the day when UV damage was highest. Cell division timed to avoid vulnerable periods. Their entire metabolism became a temporal symphony, different instruments playing at different times of the daily cycle. This temporal organization solved a fundamental problem: how to run incompatible chemical processes in the same cell. You can't simultaneously reduce and oxidize the same compounds. You can't build and break down the same molecules. But you can do one during the day and the other at night. Time became another dimension for organizing cellular chemistry, as important as physical compartmentalization. As days stretched to 15, then 18, then 20 hours, new strategies became possible. Longer periods of continuous light allowed for more complex photosynthetic processes. Longer nights allowed for processes that required extended darkness. The middle ground, dawn and dusk, became extended transition periods where unique chemistry could occur. Some organisms specialized in these transition times. Even today, there are bacteria that position themselves at very specific depths in the water column to experience exactly the right light conditions at exactly the right times. They've turned the daily cycle into a resource to be exploited with precision. The evolution of eukaryotic cells added new layers of temporal complexity. These cells, with their internal organelles, could run different clocks in different compartments. The nucleus might follow one rhythm, the mitochondria another. This allowed for incredibly sophisticated coordination of cellular processes, all dancing to the beat of the lengthening day. When multicellularity evolved, temporal coordination became even more critical. Cells needed to synchronize their activities, divide at the same time, differentiate in sequence, respond to signals in concert. The daily cycle became a master clock that all cells could reference, ensuring coordination across entire organisms. Plants became particularly sophisticated timekeepers. They had to balance photosynthesis during the day with respiration at night. They evolved mechanisms to store energy during light periods for use in darkness. They learned to anticipate seasonal changes by measuring day length, using the ratio of light to dark as a calendar. The evolution of eyes added yet another dimension to biological timekeeping. Suddenly, organisms could detect not just the presence or absence of light, but its quality, its direction, its subtle changes throughout the day. The deep blue of dawn triggers different responses than the red-shifted evening light. Shadows provide information about time of day. Biology became exquisitely sensitive to the nuances of the daily light cycle. Animals evolved their own sophisticated rhythms: migration, mating, feeding, all timed to daily and seasonal cycles. Nocturnal species evolved to exploit the cooler, darker nights. Diurnal species specialized in daylight activities. Crepuscular species found their niche in the twilight hours. The 24-hour day became divided into temporal ecological niches as distinct as spatial ones. Sleep might be one of the most profound adaptations to the daily cycle. It's not just rest, it's a state where entirely different biological processes occur. Memory consolidation, cellular repair, toxin clearance from the brain. Sleep is so important that virtually every animal does it, despite the obvious vulnerability it creates. The daily cycle forced life to evolve a two-phase existence, wake and sleep, each with its own biology. But here's something remarkable. Life didn't just adapt to the daily cycle, it began to depend on it. Organisms raised in constant light or constant darkness often sicken and die. The cycling itself became essential, hardwired into the most fundamental processes of life. We're not just adapted to live on a rotating planet, we're adapted to need that rotation. The tidal cycles added another layer of temporal complexity, especially for coastal organisms. As the moon retreated and days lengthened, the relationship between daily and tidal cycles constantly shifted. Organisms had to track both, predicting not just when the sun would rise but when the tide would turn. Some developed dual clocks, circadian for the solar day, circatidal for the lunar day. Even at the molecular level, the lengthening days left their mark. Proteins evolved that were stable for specific durations. Chemical reactions were tuned to complete within certain time windows. DNA repair mechanisms became scheduled to coincide with maximum damage periods. The 24-hour day isn't just experienced by life, it's embedded in life's molecular machinery. Part 13. The First Catastrophe. Every paradise contains the seeds of its own destruction. For 1 billion years after life began, Earth's early organisms lived in a relatively stable world. The heavy bombardment had ended, the oceans had formed, chemistry had become biology. Life was simple, single-celled, anaerobic, but it was thriving in its own modest way. Then life did something that would happen again and again throughout Earth's history. It poisoned itself with its own success. We call it the Great Oxidation Event, but that clinical name barely captures the magnitude of what happened. It was Holocaust, revolution, and renaissance all at once. The waste product of one group of organisms, oxygen, became a poison that would kill most life on Earth. But from that devastation would come the energy source that would power all complex life to follow. It was Earth's first self-inflicted mass extinction, and it nearly ended the experiment of life entirely. The villains, or heroes depending on your perspective, were the cyanobacteria. They had evolved something unprecedented: oxygenic photosynthesis. While other photosynthetic organisms carefully extracted electrons from sulfur compounds or iron, producing relatively benign waste products. Cyanobacteria went straight for water. They ripped it apart molecularly, keeping the hydrogen and discarding the oxygen like industrial waste into the environment. At first, this wasn't a problem. Oxygen is reactive. It wants to combine with other elements. The early Earth was full of reduced compounds that would eagerly soak up any free oxygen, dissolved iron in the oceans, methane in the atmosphere, sulfides in the rocks. For perhaps hundreds of millions of years, cyanobacteria pumped out oxygen that immediately disappeared into chemical reactions. They were filling a nearly bottomless sink. But cyanobacteria were too successful. They spread across the sunlit surfaces of the oceans. They formed mats in shallow waters. They colonized any place where light and water met, and they kept producing oxygen relentlessly, continuously, for millions of years. Eventually, inevitably, the sinks began to overflow. The first signs of crisis probably appeared locally, pockets of water where oxygen accumulated faster than it could be absorbed. For the anaerobic organisms living there, it would have been like their environment suddenly filling with poison gas. Those that could flee did, those that couldn't died. The cyanobacteria, protected by their own antioxidant defenses, expanded into the emptied ecological niches. But local crises became regional, then global. Somewhere around 2.4 billion years ago, free oxygen began accumulating in the atmosphere. Not much by our standards, perhaps 1 to 2% compared to today's 21%, but enough to fundamentally change the planet's chemistry. The methane that had kept Earth warm despite the faint young sun was destroyed. The planet plunged into a series of ice ages that may have frozen the oceans from pole to equator. For an anaerobic life, there was nowhere to run. Oxygen was spreading everywhere, in the air, in the surface waters, even penetrating into previously safe sediments. Organisms that had thrived for over 1 billion years found themselves refugees on their own planet. They retreated to the last oxygen-free zones, deep sediments, stagnant waters, the guts of other organisms. Most didn't make it. We'll never know how many lineages went extinct during the oxygen catastrophe. Their remains dissolved and recycled long ago. The cyanobacteria themselves weren't immune to the crisis they created. Oxygen is dangerous even to those who produce it. They had to evolve elaborate mechanisms to protect themselves. Enzymes to neutralize reactive oxygen species, repair systems for oxidative damage, compartmentalization to keep oxygen-generating processes away from oxygen-sensitive ones. They became armed camps, wielding a weapon that could kill them as easily as their enemies. But evolution never wastes a crisis. Some organisms evolved not just tolerance but exploitation. They developed aerobic respiration, the ability to use oxygen to extract energy from food far more efficiently than fermentation. It was like discovering fire in a world that had only known cold. Suddenly, there was energy everywhere for those who could use oxygen instead of fearing it. The efficiency difference was staggering. Fermentation might extract 2 molecules of ATP from a glucose molecule. Aerobic respiration could extract 36. It was an 18-fold increase in available energy. Organisms that could use oxygen didn't just survive, they thrived. They grew faster, reproduced more, outcompeted the anaerobies wherever oxygen was present. This created a new kind of world, stratified by oxygen. Surface waters became the domain of aerobies. Deep waters remained anaerobic. The zones between became battlegrounds where different metabolisms competed. Gradients of oxygen concentration created opportunities for organisms that could exploit the boundaries, using different metabolisms at different times or places. The mineral world was transformed. Iron that had dissolved freely in the oceans for billions of years suddenly precipitated as rust, creating the banded iron formations we see today, layers of red and black that record the pulse of oxygenation. New minerals formed that had never existed on Earth before. The color of the planet itself changed as oxygen reactions painted the rocks in new hues. Climate systems were disrupted. Without methane's greenhouse warming, Earth froze. The Huronian glaciation lasted for hundreds of millions of years. Ice reflected sunlight back to space, cooling the planet further. It's possible that Earth became a complete snowball, frozen from pole to pole, with only a few refugia where life persisted. Yet life did persist. In hot springs heated by volcanic activity, in cracks in the ice where sunlight could penetrate, in the deep ocean where volcanic vents provided energy, These refugia became evolutionary laboratories, places where life could experiment with new solutions to the oxygen crisis. Some organisms evolved to switch between metabolisms, aerobic when oxygen was available, anaerobic when it wasn't. Others formed partnerships, with anaerobes and aerobes living in close association, each creating conditions favorable for the other. The foundations of symbiosis were laid during this crisis, born of necessity in a poisoned world. The recovery, when it came, was spectacular. Organisms that survived the oxygen catastrophe were hardened, versatile, capable of exploiting the new high-energy world. They diversified rapidly into empty ecological niches. New forms of photosynthesis evolved. New metabolic pathways were discovered. Life became more complex, more energetic, more creative. The eukaryotic cell might have been born from this crisis. One theory suggests that an anaerobic archaeon, struggling to survive in an oxygenating world, engulfed or partnered with an aerobic bacterium. The bacterium became the mitochondrion, providing efficient energy production in exchange for protection and nutrients. It was a merger born of crisis that would eventually give rise to all complex multicellular life. The oxygen crisis also created the ozone layer. In the upper atmosphere, oxygen molecules were split by UV radiation and reformed as ozone. This created a shield that would eventually allow life to colonize land. The poison became protection. The waste product became the foundation for Earth's future biodiversity. But perhaps the most important lesson of the oxygen catastrophe is about life itself. Life is not fragile. It's remarkably resilient, but it's also dangerous, capable of transforming planets in ways that threaten its own existence. The oxygen crisis was the first time life changed Earth so dramatically that it nearly destroyed itself. It wouldn't be the last.